Hi, I am Mrs. Sloan, and this is going to be Chapter 12, Part 2. So this is Video 2. Video 1 for Chapter 12 for us was all about the history of DNA, DNA structure, and DNA replication. So here in Part 2, and this is all underneath the heading of APBio Unit 6, that's what we're on right now. And we're going to be discussing transcription and translations. Let me get in presentation mode here for you. All right, so quick overview, and let me get a pointer. Okay, quick overview. Transcription is how you use the DNA to form RNA, and we're going to talk about the different types of RNA. And then translation is how you use that RNA to build a protein, to build life. So let's get started on this. Before we get very far, I need to review with you the differences. I'm all over the place. The differences between DNA and RNA. And of course, I cannot make it there. Let me make myself smaller. There we go. The differences between DNA and RNA. So this should be a review if you're watching this video. So DNA is a double helix. RNA is a single strand. DNA has the sugar deoxyribose, and ribose is found in RNA. Look, right down here, see OH, and over here, deoxyribose. They have all the same bases except DNA has the base thymine and RNA has the base uracil. So we already looked at in DNA about how you have complementary strands, right, that hold them together by hydrogen bonds, adenine with thymine and guanine with cytosine. RNA is single-stranded, so you don't see that normally except for when it's getting built and you have that complementary base pairing. So everywhere you would want to put a thymine, instead you're going to put a uracil. And like thymine, it is a pyrimidine, which means it has one ring, whereas cytosine, guanine, and adenines are purines, right? Or sorry, guanine and adenine are purines because they have two rings to make up that nitrogenous base. Cytosine is a pyrimidine, right? So a purine is always paired with a pyrimidine. All right. Again, if you look at here, you can see what a nucleotide is. It's a base, a sugar, and a phosphate. And if you look through the nucleotides here, we have uracil and we do not have any thymine, and the sugar is ribose. So on your notes, and by the way, my notes are in the descriptor of this video that I provide for my students. Scaffolding, column 1, you fill in the notes. Column 2, I encourage you to put in pictures that are helpful. So underneath where it says, I'm on the notes, 12.3, the genetic code of life, and RNA carries the information, and you want to fill in RNA has ribose instead of deoxyribose, RNA has the base uracil instead of thymine, and RNA is single-stranded. All right, now next, here's another diagram. And just take a moment to review. I wanted just to show you one more time. It's important. All right. And that might be good for to add into your notes, the differences between DNA and RNA. Here, I'll make myself smaller so you can get the full picture. All right, so there's 3 types of RNA that you would form. So there's mRNA, tRNA, and rRNA. So M stands for messenger, and it is a coding, ultimately a coding sequence. It's going to code for a protein. Ribosomal RNA with proteins makes up a ribosome, and this ribosome, the RNA in this ribosome, acts as a ribozyme. It's a catalyst. It's RNA acting as a catalyst to facilitate a peptide bond between amino acids. So we've always talked about it being the workbench of protein synthesis. So it's got a place to hold mRNA and up to 3 tRNAs simultaneously. What's a tRNA? tRNA stands for transfer RNA. Now notice it's kind of folded back on itself with some loops, but it's just one strand of RNA in here. And what it transfers is an amino acid. And remember, you hook amino acids together to build a protein. So that's the connection, right? DNA is going to code for the mRNA. The mRNA is going to go on the workbench of the ribosome, and tRNAs are going to come to that workbench to put together amino acids to build a protein. So that's how you get it from nucleic acid language to protein language. So on your notes, let's go. On the three types, you have mRNA carries the code from the DNA, the coding sequence. tRNA carries the amino acid, the non-coding sequence. And rRNA is the workbench for translation, also a non-coding sequence. So here's a picture from my student's textbook. I want to remind you of something. Remember how DNA is anti-parallel, right? It runs from 3 to 5 and 5 to 3. And remember how we said when you replicate DNA, you always use the template strand. You're reading from 3 to 5, making complementary DNA from the 5' to the 3' end. Well, since RNA is single-stranded, you only need the leading strand. So you're going to read the DNA, right, from the 3 to 5' end, making 5 to 3 RNA. So you can see that right here on this messenger RNA. You can see the 5' end and the 3' end. Here on this tRNA, here's your 5' going around here to your 3' end. And then here, you can't really see them identified here in the ribosome, but they're there too. All right. So remember, and here's that picture I was going to show you. Here's the DNA. Remember, we talked about template and the non-coding strand. And look at the base pairing in the DNA. If you were going to make more DNA, this DNA would unwind, unzip, and both could be used to make more DNA. But when you're doing transcription, you just use the one strand, right? Because you're making single-stranded RNA. So if you look here, look up at the top, here's your DNA double helix. It will still unwind and unzip, but here you are making mRNA. And that mRNA needs to be processed into a mature RNA, and I'll show you how we do that. Once it's processed, it will leave the nucleus if it's a eukaryotic cell, and then it will begin translation. And here you can see the ribosome kind of in light gray back behind. Here you can see the tRNAs. Now, this tRNA has a chain of amino acids on it. And long story short, spoiler alert, the next tRNA is going to land, and this guy is going to dump his chain of amino acids onto the new guy and split. And the ribosome is going to shift. That's called translocation. Then the next tRNA is going to land, and he's going to get dumped on until you come to a stop codon. And I'll be explaining all of that for you. But on your genetic code intro, transcription, the process of making the RNA from the DNA. So you want to put this in your notes underneath genetic code. Process by which an RNA molecule is produced based on a DNA template. Translation is what's occurring outside of the nucleus. The mRNA transcript is used to generate a chain of amino acids in a protein. It converts the nucleic acid into a protein language. Now, if you're a prokaryote, transcription and translation is still taking place, but remember the DNA is in that nucleoid region, and they don't do the processing that we do in prokaryotes, and so translation can begin immediately after transcription because they're all in the same room. So these processes called the central dogma. It's kind of what you base the information pathway on. So DNA to making more DNA. And when would that occur? Do you remember? When you're getting ready for either mitosis or meiosis, right? For cellular division, that's when you're going to do DNA replication, when you're getting ready for mitosis or meiosis. Transcription is when you use the DNA to make any kind of RNA. It could be mRNA, tRNA, or rRNA. That is all called transcription. And where we use DNA polymerase to make more DNA, we're going to use RNA polymerase to make more RNA. Now, when you make this RNA, you're not going to copy all the DNA, right? Because the DNA is the code book. So it would be if I took this book right here and I said, okay, here is all of your DNA, right? And if I made another whole one of these, that would be DNA replication, another pink notebook, right? But transcription, when you do this, you're taking a specific page within this book and you are making an RNA copy of that page. And then you might modify it or process it. And then you use this information on a ribosome to build a protein. So you're not copying the entire book, just a section of it when you do transcription. And then translation is building that protein. So underneath central dogma, DNA to RNA to protein. So Next, okay, I wanted to show you, give you a little bit of practice in case you were asked to identify what the complementary base pairing would be. So here I am showing you a leading strand of DNA. Okay, I am only showing you the nucleotides, this portion right here, but I want you to remember that nucleotide is attached to a sugar, which is also attached to a phosphate. So if we were going to do DNA replication, what I'm asking for up here is what would be the letters that would be complementary to this DNA? And you might hit pause just for a minute. For my kids, there's probably going to be an ed puzzle question here. Identify the complementary base pairing for replication to make DNA. All right, so if you did that, I kind of scribbled it here for you. C with G, A with T, T, A, right? This all makes sense. Did I do a good job? Okay, this last one is a C. That's if we were going to do DNA replication. What if we use the same DNA strand, this leading strand here, to make mRNA? What would be the complementary base pairing to make mRNA? Now remember, what's the difference between DNA and RNA, right? RNA is single-stranded. RNA has ribose instead of deoxyribose, and it has the base uracil instead of thymine. So everywhere you would want to put a thymine, right, which normally would bind with adenine, be complementary to it, you would put a uracil. So you might pause again and see if you can come up with the right sequence here. Okay. So it would be G with C, A, U, T, A, because adenines are still here, right? This is the DNA, this is the RNA. A with U, hopefully that, oh, this is funny. It defaulted to an O. This should be a C, okay? G with C, not some funky O. Now, every 3 bases on this mRNA is referred to as a codon. So in this next one, I circled the codons for you. Okay, and this mistake is still here. So every 3 bases is referred to as a codon. These codons are going to be complementary to an anticodon on the tRNA. So remember, when you're making tRNA, this is just more RNA. So G with C, U with A with U, because it's still. So see if you can identify the 3 anticodons that would be complementary. Giving you a chance. Okay, so these, notice G with C, U with A with U. This would be referred to as an anticodon. Remember, T stands for transfer. What is it transferring? An amino acid. Okay, these are the anticodons that are complementary to the codons, and the codons are complementary to the DNA. Now, if you wanted to, you could note, look right here. CAU in the tRNA, look, CAT cat in the DNA, right? Because you're doing complementary to the complementary, right? So you can see that pattern there. Now, the codons is the code. And scientists named Nuremberg and Mackay discovered the universal genetic code that we have, because each of those codons codes for something specific, a specific amino acid, or it might be a start or stop signal, but it codes for a very specific amino acid. So you could look up GUA and know exactly what amino acid this tRNA is carrying. So you always look up the mRNA, you look up the message in order to figure out the amino acid. All right, so that here, oh, here's Mackay and Nuremberg, the scientists who did that. And here is that genetic code. I'll move myself over here. All right, so this column 1, and I'll show you a couple of different charts so you can see how it's read. Column 1 is the first base. Then these columns would be whatever your second base is, and this is your third base. So for instance, if your codon is CGA, could you look up what amino acid would code, would be brought to it? If the codon was CGNA, what would the tRNA be carrying? What amino acid? And I'll give you a second. Okay, so I would go C, right? And then the second nitrogenous base is G. So now it limits me, right? I'm in the CG. And then the third one is A. CGA is arginine. Now I want you to notice, if you had a mutation and you had CGU or CGC or CGG, you would still get arginine. And that's what's part of what's called the Wobble hypothesis, that there is redundancy. There's 4 different ways identified right here for you to get arginine. And so even if there's a mistake, hopefully it will still be okay. Those first two amino acids are really the kicker, the ones that are most important in that. So on your notes where it says finding the genetic code, Nuremberg Matai, use synthetic RNA, you have the rest of that. So let's look at the key features. I think I made a slide for you. I did. Okay, so the key features here is the code is degenerate. And degenerate doesn't mean like a degenerate, like a bad person. Let me show you what degenerate means. Degenerate refers to the redundancy of the genetic code, that if you had that mutation that I told you about, that there's multiple ways. You could get those 4 ways we looked at to get arginine, but look, here's 2 more ways to get it, right? Proline, here's 3 ways to get it. The exceptions to this degenerate or redundancy of the genetic code is right here with tryptophan. There's only 1 way to get tryptophan. It's your Ugg boots, 1 way to get tryptophan. And then the initiation or start codon is AUG. There's only 1 way to get methionine, is AUG. So that's an exception. And then yeah, I think that's what I wanted to show you there. So the first one on there, the code is degenerate. It has redundancy, more than 1 way to code for many amino acids, so it protects it against mutations. The second one is it's unambiguous. Each codon has 1 meaning. There's only 1 interpretation of each codon. And you can add that to your notes. And then there are start and stop codons. So the start codon is right here, AUG, only 1 of those. And there are 3 stop codons, 3 stop codons. So start and stop signals. This genetic code is universal. It's found in all life. And it's one of the key features for the evidence of evolution. There are a few exceptions, but it is evidence, and you want to put this in your notes, of common evolutionary heritage, meaning all life speaks that same language, that same nucleic acid language. Okay, and there's a few modifications here. All right, now, let's talk about the big picture. In transcription, right, you have double-stranded DNA. You're going to use the leading strand, and you're going to read the DNA from the 3' to the 5'. Remember, the 3' is referring to when you count the carbons in the sugar. Sugar 1 is connected to the nitrogenous base. Then you go down and then over to 2 and 3, like the bottom of the house, right? So you're reading, I'm just trying to refresh your memory on that. So you're reading the DNA from 3 to 5, but when you make your mRNA, the head of the mRNA, it's going to read from 5 to 3. And you can see they're bringing in those nucleotides. And the enzyme that's going to do that is referred to as RNA polymerase. So if we look at the DNA itself, I want to remind you that some can be coding and some can be non-coding. Coding means it codes for a protein. Non-coding means it could code for tRNA, or it could code for an rRNA, or it could be failed attempts at evolution, or it could be regulatory if it is non-coding. So this is a gene, and this is some foreshadowing to something I'm going to teach you in just a couple slides. Do you see where it says introns and exons? Exons are expressed, that's why they're called extrons. Exons, introns are actually cut out by these things called spliceosomes that actually snip out sections of the mRNA message. So here's your DNA. When you make an mRNA copy, when you do that, you will cut out the intron portion. And I'll talk to you a little bit more about that in just a minute. Okay, so transcription is using the information, the sequence of bases on the DNA in order to form your RNA. And here you can see the enzyme that does that is RNA polymerase. Now, how does it know where to start, right? Remember we said we're not going to copy the whole book. We just want to copy a gene or 2 out of this book. So the start, where you start, is called the promoter. And literally, RNA polymerase binds to the promoter. And in our next chapter 13, we're going to talk about how controlling this promoter controls what genes are expressed and what traits you have, right? So this is the biggest point of control is controlling that promoter and whether RNA polymerase can bind to that promoter. So on your notes, we are now on 12.4, the first step, transcription, which occurs in the nucleus in eukaryotes. Messenger RNA is produced, DNA unwinds and unzips, and then RNA polymerase, an enzyme, right, adds complementary RNA nucleotides to the 3' end of the DNA. So it's reading 3 to 5, making 5 to 3. The promoter is the start of transcription. It's where RNA polymerase initially binds to the DNA, and that's called initiation. That makes sense, right? And here you can see the RNA polymerase, you can see the DNA, the direction of transcription. So it's reading 3 to 5, making somewhere on this RNA is 5', and then this would be the 3' end. A summary of the 3 big steps, right? Initiation is getting this, the RNA polymerase bound to the promoter. Elongation is making your later is making your long strand of your mRNA, and then termination is when you're done with that process. All right. Now, you can have many RNA polymerases all working on the same exposed DNA strand. So the DNA unwinds, unzips, and you have a promoter, right? And the RNA polymerase binds and it starts moving along, making its mRNA. Another RNA polymerase can bind right on it, and another one, and another one. So right here in the middle would be this running like it. This looks like a sideways Christmas tree or Hanukkah bush here. This would be the star going on top here. But here is the DNA strand here in the middle. And these RNA polymerases are all moving along here. And what you're seeing dangling out here, these are the mRNAs that are getting made. So I can tell because they're shorter here on the left side of the screen and longer on the right side that it's working in this direction from left to right, because these strands of mRNA are much, much longer. So I can't remember if I told you this or not, but do you have then the promoter is the start of transcription? Yes. Elongation as the RNA polymerase reads down the DNA template, and termination, the stop sequence. And when you've stopped at the termination, you now have what's called pre-mRNA that is going to need to be processed. And I'll be talking to you about how we go about doing that. All right, so mRNA processing, the first thing you have to do is put on a cap on the head, and that cap is called 7-methylguanosine. So if you look right here, here's the RNA, mRNA coming off of, here's the DNA you can see in the background. This big purple thing is the RNA polymerase. So right here, you're going to put your cap. Remember, you're reading 3 to 5, right? Making 5 to 3. So the cap is going to go on that 5' end. Then on the tail, you're going to put a series of adenines that indicates the end of that mRNA. And then you're going to cut out those introns we talked about, leaving your exons. Your exons are in dark orange here, and you're going to remove these introns, and it's not going to happen magically like it looks like in here. It's like, oh, folding and leaving. There's a process to do that, and that process you use spliceosomes, and I'll show you that here in just a minute. Then when the mRNA leaves, this is mature RNA that can go out and hook up with a ribosome and begin getting translated into a protein. All right, and the parts that are going to get translated are the exons. They're going to get expressed, and the introns are going to be removed. So on your processing in eukaryotes, you have a cap, 7-methylguanosine placed on the 5' end, and a tail, which is about 150 to 200 adenines. And B, introns are non-protein coding sequences, and you need to fill in removed. So exons, the coding sequences remain. Okay, and in higher eukaryotes, this is done by spliceosomes. So let me show you that. I think I have one right. Yeah, so this is showing you some spliceosomes in here. The orange is RNA polymerase, and then this mRNA is coming out here. These spliceosomes are going to modify your mRNA, and they are removing the introns. So what is a spliceosome? It's small nuclear RNAs, and they're often called, when they're bound with proteins, are called snurps. And this is a place because they're cutting out DNA and catalyzing a reaction there, right, to remove some of those nucleotides. It's called a ribosome. So small nuclear RNA plus these proteins are called snurps, all right? Snurps. On your notes, I think I gave you, let's add that in. Okay, RNAs, so you want to add this in higher eukaryotes. This is done by spliceosomes, which contain small nuclear RNAs. And to identify the introns, you need to fill that in on your notes, to be removed and are cut out by a ribosome, an RNA-based enzyme, So what are the functions of these introns? Well, the first huge function is variation, because you can have one strand of DNA, and here, if we cut out these two introns right here and splice these three together, you could think of it as like exon 1, exon 2, exon 3. That would then be one type of mRNA, which would give you one type of protein, potentially. More on that later. But if you remove these two introns and this middle exon, then that would give you a totally different RNA and a totally different protein. So depending on what you cut, what you leave in, and what you cut out, that leads to variation. So function of introns, it allows for alternative mRNA splicing, which increases variability. Okay, and it may give rise to what's called microRNAs. And sorry, trying to find a spot to go. What are microRNAs? These can go back and be regulatory in which mRNA is expressed, because remember, RNA is single-stranded. So if you have some mRNA and then you have a little small microRNA, it can bind on to the RNA in that one little portion. It could be double-stranded, which would interfere with the translation of that mRNA into a protein. So we'll talk a little bit about microRNAs as regulators later. And also those introns can give rise to locations of crossing over. So on your notes, some give rise to microRNAs, which are small molecules involved in regulating the translation of mRNAs by binding and blocking their translation. Regulating the translation of mRNAs by binding and blocking their translation, and may also encourage crossing over in meiosis. All right, and good. Okay, and I gave the rest of that in your notes. I think we're good on that. All right, so now we're ready to move on to translation. And this is going to occur in the cytoplasm of eukaryotic cells. I guess cytoplasm of prokaryotic cells, but they don't have a nucleus, right? So when they transcribe, they'll immediately translate in prokaryotes. So once you have your mature mRNA, remember what I showed you earlier, every 3 bases on here is referred to as a codon, and that will be complementary to an anticodon located on the tRNA. And the T stands for transfer. What is it transferring? An amino acid. All right, and then look right here, look at AUG. Do you remember how I told you the first codon is the initiation codon and it codes for methionine? So the very first amino acid on here would be right here, and that guy where the red dot is would be methionine. All right, so the role of transfer RNA, this is 12.5 in your notes, transfer amino acids to the mRNA that is on the workbench. The codon is every 3 bases on the mRNA. And then the anticodon, I wanted to re-remind you of the codon chart, every 3 bases on the mRNA. And then the anticodon is in this loop right here. Can you see how there's a few loops on our tRNA? This loop right here is the anticodon. Notice it's GAA, and here on your mRNA is CUU. That would be complementary, right? And so we could go back and look if the mRNA, this codon right here is CUU. So let's go back one right here. We're looking up that mRNA, CUU, C, and then the second one is U, and then the third one is U. See how it says leucine? We looked up the mRNA, and look right here, amino acid leucine. So this mRNA dictated the tRNA, which dictated the amino acid. And who dictated the mRNA, right? So the DNA ultimately dictated the sequence of amino acids in all of your proteins, right? And that is how your genes are expressed. So on your notes, anticodon is 3 complementary bases on the tRNA. And then I talked to you earlier about the Wobble hypothesis, and that third base, right? We can have some flexibility there. And so even if the DNA is mutated a bit, because of the wobble in the third base of the codon, you can still probably get the same amino acid even if you have a mistake. And I gave you all the notes for that on that one. And then the next part is to attach an amino acid on the tRNA requires energy, which would be what's the energy currency of our cell? That's ATP. So here you have an enzyme that is going to catalyze. Notice here's the correct amino acid needs to bind here. You've got to get a phosphate here from the ATP, and you need to then attach on that, and 2 phosphates actually, and then you can attach on that particular amino acid and activate it, so it's activated with energy from ATP. Okay, with energy from ATP. All right. And I think I gave you that on your notes. Sorry. Yeah, requires energy. And a functional tRNAs are called charged. That's for your note. The functional tRNAs are called charged. So then let's talk about ribosomal RNA. A ribosome actually consists of a small and large ribosomal subunit. And the first thing the mRNA hooks up with is the small ribosomal subunit. And you can see in this diagram right here, the different RNA and the different proteins for each of these. Okay. And then this right here, remember, is the workbench for protein synthesis. So the structure of a ribosome, rRNA, is non-coding and it's packaged with proteins. There are 2 subunits, small and large, and it is referred to as a ribozyme because it is an enzyme now. And the function of that ribosome is, on your notes, there are a total of 4 binding sites, 4 binding sites. So 1 binding site is for this mRNA, this ribbon that you can see through here. And then 3 tRNAs can bind simultaneously. They're only showing you 2. I want you to see these sites right here. Do you see where it says A site? Okay, and then P site. And then if they showed it on here, there would be an E site as well. And E stands for exit. And so what happens is every time a new guy lands on the correct codon, right? It has to be complementary binding between the codon and the anticodon. Then the guy that's here in this P site is going to dump his chain of amino acids off into this guy in the A site, and the ribosome is going to shift, and the guy in the A site moves to the P site, and the P site goes to the E site. So this ribosome shifts. So where the tRNA lands, it would be in the A site, and then the ribosome shifts and it's in the P site, and then the ribosome shifts and it's in the E site. So every tRNA will be in a different site along that ribosome over time. Okay, so once you've made your protein, remember, it's not done yet. We have to, remember we talked about primary, secondary, tertiary, and quaternary structure. So primary structure is a sequence of amino acids. Secondary structure is an alpha helix or a beta pleated sheet. Tertiary structure is the folding pattern imposed upon the secondary structure due to the R groups, right? The R groups determine that. And then the quaternary structure is if you have more than 1 chain. So you have proteins whose job it is to get it in the correct tertiary, potentially quaternary structure. So let's see, let's go to this next slide. Also, and I'll go over those sites that are in your notes. Don't worry, I'm coming to that back. I'll circle around to that again. This is what's called a polyribosome. It looks like a bunch of mice and mice tails. But what's happening is these ribosomes, they're starting here on the 5' end, right? Remember, we read the DNA 3 to 5 made 5 to 3. And so you can see a small and large ribosomal subunit. And this ribosome, if you think of this, it would then keep moving and keep shifting, and more tRNAs would land. And then what looks like the tail on this mouse, these are the polypeptide chains. And so that's what you're looking at with this electron microscope image right here is these are the ribosomes moving along the mRNA, and then these are the protein chains that are going to need to get folded, and that is called a polyribosome. So on your notes, let's go to the A site. On your notes underneath function of the ribosome, the A site is for the incoming tRNA. The P site is for the tRNA carrying the growing protein. I'll show you this again. I promise I'll circle around. And it will pass the chain to the incoming tRNA, and the E site is the empty or exit site. Chaperone proteins begin folding the protein product, and a polyribosome is the mRNA and multiple ribosomes. So let's look at those actual steps of how that takes place. First, we'll do this overview, okay? We can see the direction of translation. Do you see our mice right here? Okay, and the tail is like the protein, and the mouse here is like the small and large ribosomal subunit growing. So remember, every 3 bases are called a codon. And so what happens is this guy right here would be considered in the P site. And I like to think of it as the protein because that's where the long protein is. And then the new guy would land in the A site, which I like to think about as an amino acid site, though it's got another name. And as the ribosome shifts, as the mouse shifts and over here to the right, then it's going to dump this chain on the new guy, and then it's going to exit like what you're seeing right here. All right, so let's take a look here and break it down step by step. Okay, so here's your small ribosomal subunit. Your very first tRNA lands directly in the P site because there's nobody there, right? The very first tRNA lands there. Notice your start codon we mentioned earlier, AUG. Here's that amino acid that tRNA is carrying, right? So small ribosomal subunit, then the mRNA, then the large ribosomal subunit binds on, okay? Look at your sites. Nobody here in the A site yet. 1 tRNA in the P site and nobody here in the exit site because we just landed. And this is called initiation. So on your notes, translation requires 3 steps. Initiation, bringing all the components together, And now we're going to start working in our elongation. So it's already gone for a little bit here in this picture. So here you can see, here's the codon, GUA, here's the anticodon, CAU. It already has several peptide bonds, which this ribosome catalyzed, so it acts as a ribozyme. Here is a new tRNA. He's going to land here in the A site. And then as the ribosome shifts, it'll move this chain onto the new guy. Simultaneously, it's called translocation. It's going to shift the ribosome itself is going to be moving along. And so now the guy in the A site is going to be moved into the P site, and the guy in the P site is going to be moved into the exit. So you can see that's happening right here. CAU, he's got moved into the exit site. Now he's leaving. And this guy right here, with the tRNA CUG, notice how he has been shifted because the ribosome moved. So now he is in the P site. And now it makes room for the next codon, anticodon. This guy's going to land. And this is going to continue on and on until you reach a stop codon, right? This right here is gene expression. Once this gets folded into its right shape, this is how you express your DNA. So elongation, the polypeptide synthesis occurs as the amino acids are added to one at a time to the growing chain, Okay, and it's going to continue until you reach a stop codon. And when it goes to shift, it doesn't shift it onto anything. You just have a release factor here. So here's your amino acid chain, and then the small and large subunits can come apart. And remember, you could have another ribosome right behind it, moving right along behind it. So termination, the components separate from each other as a result of one of 3 stop codons, and that is referred to as termination, all right? And then the first few amino acids in your chain are going to determine this protein's destination. Is it going to stay in the cell, or is it going to be secreted? Now remember, if you look up here at the top, going back to what we learned already, right? DNA in the nucleus serves as the template. Here the mRNA is processed. These introns are kind of light pink. They're getting cut out. Here's your mRNA. It's coming out. It has on a cap and a tail at this point. And then here you have translation occurring, right? Now, this ribosome could be right here on the rough ER. And remember, then the protein could enter right into the endoplasmic reticulum where it will get processed, right? So gene expression occurs when the gene product, the protein, is operating. It requires transcription and translation, and the first few amino acids of the protein determines if the protein belongs in the cell or it will be secreted. Okay, and then that is the end. So I hope that was helpful, and if you are one of my students, I'll see you in class.